Welcome, everyone. Good morning or good afternoon. This is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. And welcome to today's webinar presented by AICCT and the European Society for Toxicology in vitro. Both our organizations are active in promoting in vitro and in silico toxicology and have a number of programs and meetings to support scientists and work with students and young scientists in the field. I wanted to briefly give a moment for STIV President Matthew Vinken, who is on the line here with us, to talk a little bit about STIV activities and welcome you. Matthew, you should be unmuted. Go ahead.
Yeah, thank you. I hope you hear me. So first of all, also good morning or good afternoon, depending on where you are. Yeah, well, as already nicely introduced by Christy, so we are actually representing, at least I am representing STIV, the European Society of Toxicology in Vitro. So we try to be the leading organization in Europe to promote 3Rs and in vitro and in silico research in general, both educationally and scientifically. So we try to be very active in that respect. So in addition to the series of webinars that we started to organize last year together with AICCT, we also have a 2-yearly conference. The next one, as you can see here on this slide, will take place in Barcelona in June 2020. So we always work around the theme, and this year, at least next year's theme, is key enabling non-animal technologies for research, education, and testing. You can see the website address there. I really would recommend and encourage you to go and have a look at the website. You will see a preliminary program. We also started abstract submission a month ago. Well, I shouldn't be mistaken, I guess, but I think the submission deadline is the 1st of February. So have a look there. What we also do on a yearly basis is to organize this applied in vitro toxicology course. And for the Europeans among you, at least those that, try to be a European registered toxicologist, might be very interesting because we just got recognition from Eurotox for this course. So this is fully eligible for ERT, European Registered Toxicology accreditation. And the next course will take place in Brussels. It's a combination of theoretical courses together with hands-on training. And again, if you would go to the website, you will see the preliminary program and also the information on how to register. So thank you, Christy. Back to you.
Great. Thank you, Matthew. I also wanted to mention that the next webinar is available for registration. You can just go to the AICCT website here on your screen and register for that. It's a computational webinar about deep learning models and possible applications. And it will be held on December 4th. And then finally, our annual meeting is going to be in September. It's usually in September of every year. And so we're in the sort of planning stages for the next meeting. And I'd encourage you to get involved if you would like. And I also finally wanted to highlight another computational toxicology meeting. The International Workshop on QSAR and Environmental and Health Sciences is a meeting that happens every 2 years, either in the US or in Europe usually. And the theme this time is from QSAR to new approach methodologies, and really trying to link the field of QSAR to all of the integrated approaches that people are looking into today in toxicology to really advance the science. Abstract submission is open now. Deadline is December 15th, so I'd encourage you to think about attending. And so finally, I'll introduce today's webinar. I first wanted to mention a couple of things about the webinar itself. The webinar is recorded, and members of these two societies will have access to a recording of the webinar shortly after the event. Everyone will receive a PDF copy of some of the slides presented today, and so look out for that. All of your lines are muted to cut down on background noise. If you would like to ask questions, and I'd encourage you to do this as our presenter is speaking, you can enter your questions into the question box and I'll be able to read them after the presentation. You could also raise your hand in the GoToWebinar control panel. There's a little hand raising module that you can use. So our presenter today is Dr. Leo van Grunsven, and he is a developmental biologist who obtained his PhD in life sciences from École Normale Supérieure de Lyon in France. He had his postdoctoral training at the National Institute for Neurological Diseases and Stroke at NIH in Bethesda in the United States, and the KU Leuven in Belgium before he became head of liver cell biology research group, where he is currently at the Vrije University, Brussel in 2009. He currently studies the mechanisms that drive liver fibrosis. And so he's going to talk to us about that, about the pros and cons of 3D in vitro culture models. So let me go ahead and turn the controls over to you, and we can get started.
Okay. I hope everybody can see my screen.
Yes, looks good.
Thank you, Christy, for this introduction, and thank you all for joining me in for my overview of the pros and cons that are of our lab that we think have the current 3D in vitro cultures models of liver fibrosis have. And I'm going to mainly tell you about our perspective on this. Wait, I'll turn this down. Wait. No, I can't. Okay. So this presentation is from the Vrije Universiteit Brussels, which is located in Brussels, of course. And it's the Faculty of Medicine and Pharmacy. And here you see a drone picture where you can see that we're located next to the hospital. And we're actually located here at the ground level. And our focus the last 10 years or even longer has been to unravel mechanisms involved in liver fibrosis and regeneration. And the key cell type that we've been studying is the hepatic stellate cells. So as you probably all know, is that chronic liver disease is a big burden for our society. In Europe, at least 6% of all people living in Europe suffer from some sort of chronic liver disease, and approximately 70,000 people die every year from this disease. Luckily for us, it takes many years to go from a healthy liver to a fibrotic liver and eventually to a cirrhotic liver. At this point, you have heavy scarring of your liver, and it leads to all kinds of complications, not only of the liver, but also other organs. And usually a liver transplant will be your only rescue. Causes of chronic liver disease are hepatitis infections, fatty liver disease, or non-alcoholic fatty liver disease, which now represents or is present in approximately 25% of the world population. Abuse of alcohol and abuse of drugs. The current status is that in the clinic, there's no good antifibrotic drugs that can be used today. So we need to find better drugs, but we also need to find better models to test these drugs, because this is has been a problem in the last decades, that potential drugs found in vitro system or in animal models do not work very well in the clinic. So what kind of in vitro models do we have to build? Well, what we would like to do is to build, of course, a liver, which we can't, but we'll have a closer look at this. And the liver is made out of globules, like you know. If we have a closer look, the blood flows from the portal vein to the central vein. And I see my animation here does not work very well in this preview, but it flows through here. It passes the L6 and the hepatocytes, and the hepatocytes are, of course, the main detoxifiers of your liver. Also produce tons of proteins necessary for the entire body. The L6 are your blood vessels that make it possible that some compounds can go from your sinusoids into the hepatocytes because they have fenestrate. These are these small holes here. Eventually, you also have stellate cells, and this is where our main focus is. And these stellate cells are the biggest, or at least are responsible for most of the scar tissue formed in the liver if you have a chronic liver injury. So if you have a closer look here, this is a healthy liver. Stellate cells are located in the space of Disse. It's a space between your sinusoid and your hepatocytes. If you get damage to any of these compounds or exposed to virus, what you get is you get a damaged hepatocyte, which leads to an inflammatory response, but also activation of tissue-resident macrophages, which are called Kupffer cells. And eventually you get activated hepatic stellate cells, which will create a high amount of extracellular matrix, amongst others, collagens, laminins, etc. This has been mimicked in vivo in animal models. For instance, a very popular animal model is the CCl4 model, carbon tetrachloride. And after 4 to 8 weeks, if you take out the liver of these mice, then you can do serous red staining, which stains for cross-linked collagens. And you can see here scars being formed of collagen that block off parts of your liver. Initially, fibrosis is a good thing because it leads to maintenance of the architecture of the liver. But if it prolongs, then you get these huge scars, and it will inhibit your proper liver function. So although these models have been around for a long time, mechanisms that underlie liver fibrosis were difficult to study in these mice. So in '84, the first stellate cells have been isolated and put in culture. And these stellate cells, they have this very small look. They are very tiny cells with large lipid droplets, these yellow dots. And if you put these in a regular tissue culture dish together with serum, they will activate, and they show this large myofibroblastic cell type here. This is one cell, and it expresses hallmarks of activated stellate cells or fibrosis, which are, for instance, alpha-smooth muscle actin goes up, collagens go up, and also lysyl oxidases, which are cross-linking enzymes. So a bit of history, they have been a long time ago by a guy called Kupffer, which is kind of confusing since you also have the Kupffer cells. But hepatic stellate cells are identified by Kupffer almost 2 centuries ago. They're also called different names, so fat storing cell, parasitematosis cell, ethos cell, so it's a bit confusing sometimes. They have been isolated for the first time in rats, and the co-authors was my previous boss who deceased 10 years ago. It was only in '96 that the hepatic cell was given the name hepatic stellate cell, and everybody should refer to this, which is kind of confusing because HSC is also the abbreviation used for hematopoietic stem cell. So it's good to always look for stellate cells and not just the abbreviation HSC. So what have we been doing on these cells, and why do we work with 3D cultures? So the quiescent stellate cells goes through different phases to reach this highly myofibroblastic phenotype, which produces a lot of collagens. And initially, when I started with this work, we focused on how does a cytoskeleton look, how come these stellate cells, which cause not only a lot of extracellular matrix, but they also obtain a contractile phenotype. How do they do this? We were interested in that. Later on, we looked at the implication of epigenetic regulation. So we tested some HDAC inhibitors, and we could see, or we showed that this could inhibit this early phase. Later on, we showed that advanced glycation end products, which are highly abundant in Western diets, can stimulate the activation. And for instance, capsaicin, which is the ingredient that causes the spice in your food, it can block partially this activation process. But it was more the very basic studies on processes involved in the early steps like autophagy, the unfolded protein response, but also a molecule called YAP, which is a mediator of the hyposignaling pathway, that made us realize that regular tissue culture dishes or regular tissue culture of a monoculture of hepatic stellate cells were not a good way to go. And I will have to go more into that. So all these studies were done with primary mouse stellate cells, and we isolate these stellate cells by doing a perfusion of the liver. We then, after the single cell suspension of the liver is quickly spun down, and the non-parenchymal fraction, we either put in a fax and to isolate the stellate cells, or we put them on a nicodense gradient, and the stellate cells end up in this layer here. We can put them in culture, and this is how they look. What we can also do is isolate from these pellets, put those on a parenchymal gradient, and we get hepatocyte cultures. So when Inge and Natalie did this, they isolated the stellate cells from a healthy liver or from a liver that was exposed to CCL4 for 4 weeks. And they isolated by using a fax and using the autofluorescence of the vitamin A, so the yellowish droplets that you saw a couple of slides ago, they turn up as autofluorescent dots on a fax plot. So if you isolate these cells and we immediately profile them for the RNA levels, and we could compare those cells, so these are quiescent cells from a healthy liver, these are activated cells from a fibrotic liver. We could also put them in culture and have them in vitro activated. So we compared in vitro activated stellate cells with quiescent cells with in vivo activated stellate cells. And to our surprise, when we did the RNA analysis by still back then regular RNA chips, we can see that only 25% of all roughly 1,000 genes that are differentially expressed in an in vitro system, only 25% is also differentially expressed in an in vivo system, which shows us that this in vitro activation of stellate cells is not the best model to use. However, what we also noticed is that the hyposignaling was maintained in vitro and in vivo. So it would play a role both in vivo and in vitro during stellate cell activation. And the hyposignaling relies on YAP and TAS, which is normally, if the hyposignaling is on, YAP is in cytoplasm, and then it's shuttled to the nucleus and it turns on these these target genes. What we could show is that if you isolate stellate cells from a mouse and you look at them immediately you spin them down and you look at them, YAP here in green is in the cytoplasm. If you wait 10 hours, you can see that most of the YAP, or at least some of the YAP, already is in the nucleus 10 hours after culturing them in 2D. And after 10 days, all the stellate cells have YAP in the nucleus. And you do not only see this in vitro in 2D cultures, but this phenotype of YAP in a nucleus also occurs when you look at the stellate cells after 4 weeks CCL4. There's many cells, not all, but more than 50% have YAP in the nucleus, and we could also show that in human stellate cells in humans that had a hepatitis C infection. So this told us that YAP is required for, or at least is associated with stellate cell activation, and that this happens really fast during culture in 2D. And then to circumvent this rapid activation in 2D, what we did is we put them in a spheroid in 3D. And so you can see here that over time 2D cells activate and they get this myofibroblast phenotype, but in 3D they form nice spheroids. Actually, already after 1 day or 2 days, they form spheroids. And if you then look at stellate cell activation, you can see that ACTA2, which is smooth muscle actin gene, goes up heavily already after 4 days in culture here, and goes up 100-fold or more, 500-fold after 10 days. In 3D cultures, this is not the case. So you only get a 10 to 20-fold upregulation of ACTA2. And this is in line with YAP target genes, which in 2D, after 10 hours when YAP is already in the nucleus, these target genes of YAP are upregulated and then are downregulated again. This YAP target gene activation does not happen in 2D. And we could show in the same paper that if you knock out YAP, stellate cells do not activate anymore. So what was a good in vitro alternative in '84 does not seem to be a good alternative anymore, because the stiff surface where you plate the stellate cells on apparently causes YAP to go to the nucleus and immediately activate the genes, immediately activates stellate cells. Now, it activates the stellate cells as it might also do in vivo, but the mechanisms by which it does are not entirely the same, because only 25% of the genes overlap. So we think maybe 3D cultures would be a better alternative. However, in this monoculture of stellate cells, you do not have the hepatocyte damage. So we looked whether we could replace this 3D monocultures by using co-cultures, and for us, the best co-culture would be to have at least functional hepatocytes and quiescent stellate cells, to make it very simple. And upon the damage, you would get stellate cell activation, which expressed collagens and voxels, etc. And we call this an in vitro fibrosis model. We looked at different cultures of 2D, just plating stellate cells on top of hepatocytes, or putting stellate cells on the bottom and hepatocytes in a hanging insert. But it turned out that most of the stellate cells always activated. So we turned to 3D cultures, since in 3D cultures, stellate cells will not activate on their own. We had to look, or at least find out what we would start off with. Would it be best to go for stellate cells from mouse or stellate cells from human? And since we would like to have a system that would also predict what happens in the human, we compared stellate cell signature of activated stellate cells from the human in vitro with activated stellate cells from the mouse in vitro. And it turns out that even if you compare these two very simple systems, only 50% of in vitro, at least upregulated genes, overlap between human and mouse hepatic stellate cell activation. And a good example of this is mouse neurotremin, which in the mouse is a gene associated with quiescent hepatic stellate cells, which is rapidly downregulated in 10 days in culture. But in human stellate cells, this gene is very low and is rapidly upregulated in activated stellate cells. So we started to culture, or at least use, human hepatic stellate cells in 3D spheroids. And to make the story short, we use primary human stellate cells that we isolated in the lab ourselves from donor livers, and we used differentiated HepaRG cells that we obtained from Biopredic, already differentiated, and we just thawed them, put them together with passage human hepatic stellate cells at a ratio of 2:1, so 2 stellate cells with 1 hepatocyte. And the spheroids are roughly 200 micron in diameter. If you open these, you will see that most of the hepARG cells are in the surrounding, so surrounding the stellate cells that are located in the core. And if you cut these spheroids, you stain them, you can see that CYP3A4 staining is mainly located at the rim of the spheroids, while the stellate cells are located in the center of the spheroids. So if you use these spheroids, we could keep those spheroids in culture for 3 weeks or more, and I refer to the paper to get more detailed data on this, but I quickly give you an overview. So we wondered, well, can we damage those hepatocytes and then cause maybe a fibrosis? And the easiest way for us was to go for acetaminophen, because it was the best known hepatotoxicant. So if we do this, and we do this on day 20 in culture, we can see that increasing amounts of APAP give ruffling of the spheroids, and which indicates spheroid damage. We looked at ATP toxicity by measuring ATP, and we could determine EC50 for hepARG stellate cell culture, which was roughly 5 millimolar, which is very good. hepARG cultures on their own in 3D give 9 millimolar. Stellate cells alone are not really affected even in 3D. So can we induce stellate cell activation by damaging the cells with APAP? Here we see an analysis of the RNA levels of the spheroids. We pull roughly 6 spheroids, isolate the RNA, and we determined the levels of, for instance, LOXL2, which is the cross-linking enzyme that goes up during fibrosis. hepARGs alone have very little LOXL2. Stellate cells have high amounts of LOXL2 when they are in 3D culture, or at least when they're alone in 3D culture. The levels are lower when they're in 3D hepARG stellate cell cultures, and when you treat them with APAP, this level goes up. The same is true for collagen. You see only in the hepARG stellate cell culture that APAP causes an upregulation of these genes associated with hepatic stellate cell activation and thus fibrosis. So can we look at inflammation in these spheroids? Well, not really, because we don't have Kupffer cells in there, but at that time we used a cytokine mix, which is linked with inflammatory response. By adding these inflammatory cytokines, we could shift the toxicity curves to the left, indicating that at a lower concentration of APAP, we saw the same toxicity occurring. And this also is true for the RNA, at which levels the stellate cell activation took place. So this is the normal culture, and if you add the cytokine mix, the curves turn to the left. So at a lower concentration, you have a higher activation of collagen and LOXL2 transcription in these cultures. So how about repeated exposure? Because a single exposure sometimes does not do the job. So this is where we did, for instance, 4 exposures over 8 days. So this is a single exposure at the end, but if you treat for 4 times in the week preceding the detection, you can also see that your ATP curve goes to the left, indicating higher toxicity at a lower concentration of APAP. And the same thing happens for the activation of the genes associated with fibrosis or stellate cell activation, LOXL2, COL1A1 and 3A1. They all shift to the left, indicating higher activation at a lower concentration of APAP. So can we really evaluate the risk for fibrosis in vitro? Excuse me. In this biomaterials paper in 2016, we tested methotrexate and allyl alcohol, which both have been indicated to cause fibrosis in different models, let's say human and rat. And of course, our model was from the start that we induced hepatocyte injury by using APAP. And when we tried to submit this paper to biomaterials, they said, well, you know, APAP is not a very good, it's not known for its fibrotic response in animals. So we don't really believe this. So we went to the mouse, and what usually people do is they starve the mice overnight or 12 hours, and then give a high amount of APAP for 1 injection. And this causes indeed acute liver failure. But if you give a mouse normal chow, and you give a little bit less of APAP, you can also see that there is injury. So these are ALT levels indicating liver injury in these mice. But if you cause this injury in a regular mouse, the mouse does not die. At least these kind of injuries does not kill the mouse. If we then, after 1 injection with 300 milligram per kilogram APAP, which clearly does not kill the mice, if it gets regular food, and we sort the stellate cells from these livers, and we look at the gene expression profile, we can see that for both smooth muscle actin or ACTA2, collagen, and LOX, these levels are indicative of an activated stellate cell in APAP-treated mice, only after one injection. So we then went on and treated the mice as we would do treat mice with CCL4. We injected twice a week. For 4 weeks, we injected the mice with APAP, and we can see here that there's some fibrosis occurring in BALB/c mice. We quantified this, and there is activation, or there is at least fibrosis or scar tissue formation, not as much as we sometimes see in CCL4, but we do see an activation. And a C57 black 6, this was even a bit nicer, at least it was cleaner. And we could also see fibrosis. So APAP does cause liver injury, and subsequently it can cause fibrosis as well if you just take it for a long time. So I guess we could say that we can evaluate risk for fibrosis in vitro in this model because we can look at damage of the hepatocytes and stellate cell activation in these cultures. Now, what is the added value over just looking at hepatocyte damage? You might say, well, we can also induce directly stellate cells in these cultures by just adding profibrotic compounds like TGF-beta or even LPS, which activates directly stellate cells. So the title says pros and cons of these cultures, which I'd like to discuss now a bit. So what we have been using so far, and still actually, and I've been talking about in this talk, is we've used primary human hepatic stellate cells that we isolated from donor tissue ourselves in the lab and characterized them. These cells, when you put them in culture, become more quiescent, and then you can, at least when they're put in culture with hepatocytes, they become quiescent, and you can then activate them once you damage the hepatocyte. However, you're still, for the hepatocytes, sorry, for the hepatic stellate cells, you depend on donors, which at least in our hospital are not very frequent. The HepaRG is only one cell source, which of course is also not ideal. It was very good for us to optimize the model, but it's just one hepatocyte or hepatocyte-like cell, and it's not a primary cell. What I don't have, I guess, have to explain, but I can go into that, is that the spheric culture are very tedious. You have to change the medium every couple of days, every 1 or 2 days. You should not lose these spheroids when you change the medium. They're only 200 microns. And also the analysis afterwards is very time consuming, because if you want to do staining, you have to embed those spheroids, section them and stain them. RNA analysis, you have to pool cells. Even ELISAs on media, you have to pool. So you need a lot of cultures at the same time going on in your lab. A very important drawback of this culture that I presented so far is that it's only 2 cell types. So I will take you through some of the drawbacks and how we try to solve them. So one is that we depend on donor livers. We don't want to go on and always depend on this. So what we thought is, maybe we should just order some online. And actually Biopredic, who also provided us with HepaRG cells, said, well, we also isolate stellate cells. Maybe you could test some for us. And so we did. We got 4 of those cell lines from them, and we put them in culture. And here you can see that these different isolated stellate cells from different donors have clearly a different phenotype when you put them in 3D. Again, you see that the HepaRGs are surrounding the stellate cells in the core, but you can see here as well that these BP16 cells express a lot of PGF receptor beta, and the protein expression of PGF receptor beta is associated not only with stellate cell phenotype, but the more PGF receptor you have, or the higher expression, the more activated cells can be. So you can see a clear phenotype difference. We then expose the cells to either a single exposure of APAP or TAA, so thiacitamide, for 3 times, because one single exposure of TAA does not do the job. And as you can see here, HepaRG 3D cultures only get damaged, but MTA not that much. And we also do not see that much collagen staining in these spheroids. If you then take a look at our PDGF, sorry, our BP16, which had a high level of PDGF receptor beta expression, you can also see that in the controls, there's a lot of collagen expression already present. And this does not increase much if you expose to APAP. There seems to be a bit more, but it's very difficult to distinguish. And also TEA, there's high levels of collagen. If you, on the other hand, take BP17, you see very little PDGF receptor beta staining. If you damage the hepatocytes, the damage seems to be very good. And you also increase PDGF receptor staining in the core of the spheroids, and the same thing for TEA. So ordering hepatic stellate cells is good, but it's best to test before use, which is something that we're now setting up. How about not using donors anymore, but making stem cell derived stellate cells? So for this, we collaborated with Kathleen Vrije from the KU Leuven here in Belgium and Pau Sancho Bru from IDIBAPS in Barcelona. They developed a protocol in which pluripotent stem cells could be differentiated in pluripotent stem cell derived hepatic stellate cells or hepatic stellate cell like cells. And if you passage them, they become more homogeneous and they look a lot like primary human stellate cells that are also passaged. If we do RNA profiling on those, we can see that these stellate cells are located somewhere between quiescent human freshly isolated stellate cells or cirrhotic stellate cells freshly isolated or passage stellate cells. And here are BG1, so this is the fibroblast line of which these cells, the iPS cells, are derived. So it's somewhere in the middle. They're not really stellate cells, they're not iPS, they're not a regular fibroblast. So with respect to transcription profiling, they're somewhere in the middle, and we asked ourselves, well, how should iPS stellate cells behave? They should express hepatic stellate cell markers, and we show that they do. They express all these markers also present in primary cells. They should respond to profibrotic stimuli, which they did. TGF-beta or PDGF activates the stellate cells partially. They should store vitamin A in lipid droplets, and for this you require ALRAD, which is an enzyme that puts vitamin A or retinol into lipid droplets. And you can see here that iPS-derived stellate cells do express this RNA, while regular fibroblasts do not. But for us, the icing on the cake would be if the primary stellate cells would behave like the iPS stellate cells in co-culture. So we replaced the HepaRG stellate cell mixed with HepaRG iPS-derived stellate cells, and they also form nicely spheroids. Initially, you thought that the distribution was a bit different, that HepaRGs, while they normally are surrounding the stellate cells, they were more like salt and pepper, but eventually we follow-up cultures showed also that we have a similar distribution of the hepatocytes and the stellate cells in these cultures. And what we could also see is that if you look over time, so this is over time in culture after the spheroids are formed, so these are the iPS-derived stellate cells and these are the HepaRG with the primary stellate cells, you can see that collagen 1A1, which is an activation marker, actually goes down in culture, which shows that these stellate cells become more quiescent. And this is the same for the primary human stellate cells. Well, if you only put them together, so human stellate cells in 3D, this does not happen. So iPS stellate cells also gain quiescent features when you put them together with HepaRGs. And here you can see that also the HepaRG plus iPS-derived stellate cells, actually after a short dip, after the first couple of days, they regain a CYP3A4 expression and they also have good hepatocyte functionality. So when you expose these to APAP, the HepaRG iPS-derived stellate cells, these spheres also undergo some cell death, and you can see cross-linked collagen formation in these spheroids, and you see more collagen deposition in those spheroids. If you take the medium from 24-hour exposed APAP cultures, in this case at day 14, you can see that in both iPS-derived stellate cell HepaRG cultures, but also in primary stellate cell HepaRG cultures, collagen ELISAs show that collagen is increased in the medium. So we're not really depending on donors anymore. I think we can use iPS-derived stellate cells or iPS-derived stellate cell-like cells because they give us the same readout as primary stellate cells. And in the paper, we also showed that human ES cell-derived stellate cells are also possible. Then only 2 cell types. Is this a problem? Well, so far I've shown you that we can show acute liver injury, and we can also show that this damaged hepatocytes leads to activation of stellate cells, which gives you an extracellular matrix deposition, or at least collagens and oxal goes up. But there's, of course, other liver diseases which sometimes require more than just one cell type, especially, for instance, NAFLD. We would be very interested in whether we could mimic that steatosis or a fatty hepatocyte would also induce an activated stellate cell phenotype. So this is what we did, and we used the Biopredic human stellate cells, and we made spheroids with them, and we exposed them to lean and different fat media that we obtained from literature, because a lot of people do now fat accumulation studies in mainly hepatocytes, but we did this in hepatocyte plus stellate cells, and we can see that most of these fat media, especially 2 and 4, lead to a high upregulation of fat content in these spheroids. But we don't really see a stellate cell activation if we look at LOXL ACTA2 or LOXL2 or collagen 1. Maybe this FAT1 increased a little bit the activation, but we also see that there is some damage already in these spheroids. So just lipid accumulation does not seem to induce stellate cell activation. We try to combine this with TGF-beta and LPS. And there we actually see that, for instance, if you look at ACTA2, where we see an upregulation of ACTA2 in lean conditions, actually having a fatty media seems to inhibit rather than to induce stellate cell activation in these cultures. So we think that we need to incorporate macrophages in these cultures to really get a good stellate cell activation due to fat in these cultures. So 2 cell types is not enough, so this is a big drawback. So at this moment we have HepaRG and primary stellate cells, or HepaRG with 2 iPS-derived stellate cells as a culture system. So can we go more complex? And I think we can. And we have been looking into whether we could isolate all the best cells from a primary mouse, directly, and without putting them in culture, put them in spheroids. But since we didn't want to go immediately to this very complex culture, we first looked whether we could recapitulate what we saw in these HepaRG line plus primary stellate cells in culture, whether we could do this with only primary cells that have not ever been in culture. So this would be the best model possible. So we use the same ratios, 2 hepatic stellate cells towards 1 hepatocytes, and you might wonder why we use this ratio, because it's not at all what the liver looks like, but we found that hepatic stellate cells, if we put this ratio towards hepatocytes, that hepatocytes behave better, they maintain their functionality better for a longer time. If we went below a 1-to-1 ratio, clearly hepatocytes didn't behave as well. So primary cultures, not plated, not cultured at all on 2D, they form rapidly. So after 1 day you see this, after 2 days you have a nice spheroid. You don't see, by the way, you don't see that the stellate cells are in the center. They're all over the place, here indicated by Desmond. So you don't have this core of hepatic stellate cells. It's more pepper and salt. You can see that compared to 2D, at least the hepatocytes are doing better in 3D with stellate cells. And you can also see that stellate cells in 2D activate, while in 3D this does not happen for the primary cells as well. So then we looked whether we could test antifibrotic compounds in 3D mouse models. We did, of course, a lot more tests, but I think these are the most interesting to show. So we focused on, first of all, on YAP signaling. We showed in this paper that if we treated mice with CCL4 And the last week of CCL4, we also included fetoporfin. We could block fibrosis, a clear reduction in fibrosis. And if we used valproic acid, we showed many years before that valproic acid together with CCL4 does not cause liver fibrosis. So we tried to mimic this in vitro. We used these cultures after 7-day in culture and we exposed 3 times with APAP versus 1 time with APAP, and then looked at day 10. And we can see here that 1 exposure with APAP for 24 hours increases ACTA2, but 3 exposures increases even more ACTA2 in these cultures, and the same is true for collagen. If we now give VPA or verteporfin during this time period, we can block this activation completely. So it shows that what happens, at least what works in vivo, seems to also work in vitro. So then we wanted to know, can we go more complex? And for this, the regular isolation procedures we used were not that good because our spheroids did not form very well. So what we're used to do to isolate these cultures is we would do an in vivo perfusion. We would wash the cells and then come with antibodies like against LSEC, CD32, and for Kupffer cells for F480. We would isolate them using FACS. And then we put these cells together. But the procedure is rather long, so 3 hours, and these cells somehow do not like this. So a PhD student in the lab found a different system. If you use a secondary antibody into your mice 2 hours before you isolate the liver cells, actually a secondary antibody is taken up by LSEC. They have Fc gamma receptors. They take up actively the receptors, and Kupffer cells take up aggregates of these antibodies. And in the end, your FACS plot look like this. These are the LSECs, these are the Kupffer cells, and these are stellate cells, UV versus your Alexa antibody, let's say, a secondary antibody injected 2 hours before in the tail vein. And you can isolate your cells, which are very pure. We can do this from BALB/C, BLAC6, or rats. And although the plots look a bit different, you can isolate these different cell types from all these mice, put them together with hepatocytes, and this is cross sections of these spheroids, and you can see that hepatocytes, stellate cells, endothelial cells, and macrophages are present. And we're now looking to induce NAFLD cholestasis or steatose in these mice. So, of course, what we really want to go to is not use primary mouse anymore, because we would still need mice, and the mouse is also not really resembling a human that much. We would like to go all for iPS-derived stellate cells, hepatocytes, LSEC, and Kupffer cells, and I think we're well on the way to have iHep, stellate cell cultures, but Kupffer cells and LSECs, iPS-derived, are not very well established. However, a recent paper showed us differently. There's a cell metabolism paper from last summer that showed that while first going to an endodermal step in iPS cultures with a short retinoic acid step here between day 6 and day 10 in Matrigel allows them to have human liver organoids formed, and then afterwards they induce fibrosis by exposing them to oleic acid, which causes steatosis and then inflammation and fibrosis later on. They even characterize their iPS-derived organoids, and they show that in their organoids they have stellate cells, they have hepatocytes, they even have some Kupffer cells in there. And what they could do is that if they induce either fibrosis directly by LPS or with oleic acid, they could measure using atomic force microscopy, they could measure stiffness increasing with increasing amount of LPS or oleic acid. So this is a very important study in the field showing that iPS-derived co-cultures are feasible. Another recent development, and this fits more in the safe time order online, is you can use in spheroids, which are primary human hepatocytes, stellate cell, LSEC, and Kupffer cells put together in a spheroid by in Sphero. And this last paper from March this year showed that even a very simple model in which they cultured for in total 9 days in the presence of palmitic acid, they could show an activation of stellate cell activation markers like collagen 1A1, 3A1, although no SMA is increased, but they could show in ELISAs that collagen 1 N-terminal peptides, which is a pre-processed form of collagen, was upregulated threefold, as well as collagen 3 was upregulated. And in stainings, they could show cross-linked collagen here in blue in these spheroids. So this is a very nice model. And if I would say, if you have, the money, maybe in Sphero are very good, as very good spheroids for you to test these compounds using a very simple palmitic acid model. And then this is maybe a bit older. This is in 2016, where primary, sorry, I see that I almost at my 45 minutes. That primary uses primary human hepatocytes, macrophage, and stellate cells, and the hepatocytes stay very well. But it's a fluidic device. It's not micro because these are 10-centimeter dishes. And what was striking that if they expose it to a lipotoxic compound, the stellate cells would also activate. So I haven't seen a follow-up of these cultures, but I think these are also very interesting culture systems that people could envisage to test fibrosis in a dish. Although the huge amount of cells necessary here maybe would limit everything a bit. So to finish off, pros and cons of 3D cultures. I think if you use 3D cultures of hepatocytes and stellate cells, in our case was hepARGs and stellate cells, either primary or IPS derived. The positive part is that stellate cells are quiescent, hepatocytes are functional. You can see stellate cell activation after hepatocyte injury, which is something you cannot do in regular 2D cultures. And you can even test drugs that inhibit fibrosis. The negative parts clearly are primary human hepatic stellate cells, and you depend on donors or a good source, commercial, but these usually are not tested in such a system. But IPS-derived stellate cells are an option and are very reproducible. hepARGs, the use of hepARGs is maybe also a negative point, but also a positive point. It's also very stable, but it's only one cell type. And also here, IAPs are an option, and maybe the use of different IPS lines with different mutations could also give you a better insight. You could also still use primary heps, but there we've seen that not all hepatocytes make good spheroids. Then I didn't go into the spheroid cultures are tedious and analysis too time consuming. This is the way it is. For the moment, we are working on different ways to maybe increase the imaging capacity. And then maybe you could look like they did with electronic force microscopy, look at every spheroid and see whether you have fibrosis. And then last, only two cell types. I think this can be extended, definitely using IPS like this one paper showed. Or using primary mouse stellate cells, primary mouse cultures, because there I think you have the best cell types possible. And then I'd like to thank everybody from my lab and the previous people that passed in my lab as PhD or postdoc, and I gladly take your questions. This was it, thank you.
Great. Well, thank you so much for that presentation, Professor Van Grunsven. We do have time for some questions. So again, if you want to ask a question, feel free to type it into the question box, or you can raise your hand and I will be able to call on you. I think your presentation was really nice because it showed kind of the development and evolution of some of these models. And we have a hand raised, so I'm going to hold my question so that we can let our questioner ask. So let's see. Paul, go ahead.
Yeah, hi Leo. It's Paul Jennings here from the Vrije University of Amsterdam. Nice to see your talk. Nice to hear from you again. I was just wondering, because I mean, your talk was about the advantages and disadvantages of 3D culture. Is there a potential disadvantage as well about penetration of compounds? Because my feeling would be that the external cells would have a higher uptake of a compound, and then towards the center would be less. Do you have any evidence for such a phenomenon?
Well, yes, you could think this, but they're only 200 microns. And for instance, as you can see, is that at least for the hepARG stellate cell cultures, the stellate cells are in the middle, And if we just give, a bit of TGF beta, we see a very quick response. In a couple of hours, we see inhibitory SMADs go up in 2 hours time, while the stellate cells are in the core of this thing. So yes, I think there's always a problem if you have to go through a couple of layers, but it seems that growth factors do get in there, and I assume that also a compound would get in there. Because it's just 200 microns. It's not that big.
Yeah, yeah. Oh, great. Thanks.
Thanks, Paul. Any more questions? I will go ahead. I was struck actually with what you were talking about when you went to publish some in vitro results, and the editors or maybe the reviewers kind of said, well, we want to see some in vivo evidence, and that leading to some in vivo experiments. And so I was wondering, what turned out to be the case was that you were able to match the results, but in some cases, of course, that won't be the case.
No.
And maybe not, and the in vitro model may not be wrong. And so I'm just wondering if you had any thoughts about that, about maybe how scientists as a community can grapple with some of this sort of bias on the part of reviewers and journal editors about in vitro results.
Yeah, I think I think it's very difficult to circumvent, and we all have tried to publish papers, I guess. And if the reviewer says, well, your model is no good because I don't know anybody with fibrosis due to APAP, what are you going to do, right? So it's very difficult, and we could state that we've done all the experiments and you show that there's damage and you show that there's stellate cell activation. But if a reviewer states that this is not good enough for him, either you try to argue or you do the experiment, right? So it's very difficult, and it could have turned out that it was not the case, that in vivo it would not cause stellate cell activation. And I guess in this case we were lucky. Or maybe it would have taken, a year. Mm-hmm. That, but yeah, then we would not have succeeded. So I think it's very difficult that if a reviewer or even a granting organization says, well, this is not very relevant for us because we don't know of anybody having or suffering from this or that, it's very little we can do. Mm-hmm. People are not yet really that much convinced on only in vitro data, unfortunately.
Yeah, I wonder if this could be a topic for a panel discussion, talking about strategies for addressing the situation, to help authors try to counteract some of that pushback by reviewers.
Yeah. I don't know how to address this. It's a similar approach that you have. I mean, we also do some in vivo experiments still. If you want to publish a paper and you've set up your G-power analysis, everything is okay, but they want to see 3 biological repeats. Mm. While if you repeat it twice and it shows the same, and you already build up your experiment in such a way that actually everything is, is okay statistically, they still want 3 repeats. Mm. And which is nonsense. But there's nothing you can do, right? So it's the same with just doing in vitro. If you show a mechanism blocks fibrosis in vitro, they want to see the mouse model or the rat model, indicating that this is indeed the case. Mm-hmm. Maybe not if you go to only in vitro journal like in vitro toxicology. Maybe they will allow you to do this, but some other journals, more like the liver journals, in this case a biomaterial journal, also did not believe this, depending on referees, I guess.
It looks like Dr. Jenny?
No more questions?
Yeah, there's one more. Paul has another question.
Yeah, I was just going to say, Leo, we in vitro toxicology will, of course, never ask you for animal verification.
Okay. Good. I thought so.
Good to know.
Could I just ask one more question if there's no other ones?
Sure. Yeah, we don't
Sure.
have together, so go ahead.
Okay, Leo, I'm really struck because we're doing work here, actually iPS-derived podocytes from the kidney, and your images they look a lot like our cells, and it might be because both of the cells are pericyte-like cells.
Yeah.
So I'm wondering if you've ever had a look at podocytes, and just see they look very, very similar by phase contrast.
You know, like I said, they're not hepatic stellate cells yet, right? I mean, we can do a lot of tests on that. Maybe we could, you know, if we're going to do some RNA-seq on these in the weeks to come, because we only had, you know, arrays. If you have any RNA-seq of your podocytes derived from iPS, we could, you know.
Yeah, we do. Yeah. And I think they honestly might look like each other simply because they have a similar kind of origin developmentally, which is kind of cool.
And of course, you also maybe, you know, the way we culture them, they're on a flat surface. You push them also a bit to stretch, and you know, after a while, some cells seem the same. Maybe if you put them off the regular collagen-coated dish or whatever you grow them on, and you put them in 3D with some other cell types, they might not look the same anymore.
Yeah, that's true. Yeah.
Great. Well, I don't see any more questions. So thanks a lot for this really interesting post-presentation discussion. And I guess we will say goodbye to everyone. Thanks again to Professor Van Grunsven for your presentation. Thank you all for listening, and have a great day.
Thank you. Bye, everybody.
Bye-bye.